Hi, today I have interesting problem for you. You may try to solve this problem on your own, but I just want to warn you that this problem is tricky. So still you may try to solve this problem, and when you would be ready, you can run video again and you can compare your answer with my answer and explanation. So here is the problem. Whenever a capital letter is present, a red color is produced. In a cross of one parent that is heterozygous for both genes with another one that is also heterozygous for both genes, how many red offspring would you expect out of 16? And let me start with writing down the genotypes of both parents. So parent 1 would be capital A small a and capital B small b. We have to cross with another one that is capital A small a and capital B small b. And now let me demonstrate you something. For example, when we are saying that in order to produce a red pigment we need at least one capital letter for both genes. That means, for example, if we consider these alleles, that is one is capital A, another one is small a, and for example when we have a chain of reactions, and in the biology it's a very rare case when you can find that one protein would make some biological activity. Usually we need many proteins that aggregate together and work together in order to produce some biological activity. So just very few proteins work alone on their own. So imagine that such biologic pathway consisting of two events. For example, here we have one gene, and it would be transcribed into the message RNA. So this is DNA, and this is going to be RNA. And here we would have some protein produced. So this is going to be protein. And imagine that this is gene A. And now imagine that the same gene that is going to be gene small a. And here is the DNA. But imagine that this time it can be defective. For example, something is mutated here, and as a result the message RNA would be also produced defective, and when it's going to take its shape, the protein going to take a shape, imagine that this shape would be different from that shape that allele capital A would produce. So you see, these two proteins would be different because of this mutation. And in order to produce a red pigment, we need both genes A and B be normal. So for example, this is going to be gene B. It can be shorter, for example. It also can be DNA. And this is going to be capital B. And capital B would stand for the normal gene. And this is going to be RNA. And this would also produce some protein. And we also might have gene B, that is small b, and this is also going to be DNA, and here would be some mutation that makes it recessive, and also RNA would be produced with some mutation here, and that means that protein that would be produced also would be misshaped. So here would be different conformation of the same protein. And when we have, for example, this normal allele for protein A, and this normal allele for the protein B, we would have normal shape of the protein. For example, like this one and like this one. But when we have a defective protein A, that is small allele, and for example, normal allele for the protein B, we would have a misshaped protein, even if subunit B would be normal, subunit A would be misshaped, and no red pigment would be produced. And as you see, there are different combinations possible. For example, if we have capital A, and small b, no red protein would be produced because two alleles B would make a defective protein. And the same would be if we would have small a, and for example capital B, small b. Those here we have at least one normal allele for the subunit B. We don't have in such progeny any normal allele that would produce normal subunit A. So this also means that such genotype wouldn't be able to produce red pigment. And usually in plants, for example, when a plant cannot produce red pigment, that means white color. So how to find now the probability for the such a cross, like here, of these two parents to produce a progeny that would be able to produce red pigment. And this is very easy. We just have to think about these two genes and two allelic pairs. One is here and another one is here. We have to consider them separately and we have to build a Punnett square. For example, here on the top would go genotype of the parent 1 and parent 2 would go on the side. And as you see, if we build a Punnett square, we would see that probability for the normal set of genes or alleles here would be 3 out of 4. So all these 3 genotypes would produce red pigment. So this we can say that this is 3 out of 4 probability, or 0.75, or 75%. So this is 3 different types how you can notate this, and all of them 3 would be correct. And let's do the same for the gene B. So parent 1 on top and parent 2 here on the side, and on the top we would have capital B, small b, and capital B, small b here on the side. And now when we build the Punnett square, once again here we would have capital B, capital B, capital B, small b, capital B, small b here, and small b here. And as you see, once again we have 3 out of 4 chances to have normal protein to be produced by such genotypes as capital B, or capital B, small b. So once again 3/4 chances, or 0.75, or 75%. And as you see, in order to solve this problem, we just have to make a simple mathematical calculations. So we have to multiply 16, and this is a number here. We have to multiply by the probability to have normal A allele. So we have to multiply by 3/4, and we have to multiply by the probability for progeny to have normal set of B alleles, so whether two capital B alleles or at least one capital B allele should be present, and the probability is once again 3/4. And if we multiply 16 by 3/4, here we are going to get 12. And once again we multiply by 3/4, and the answer would be 9. So this would be our answer today, and this is the correct answer, would be C. Thank you for attention. This is all for today. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Goodbye.